Hello and welcome to the AQA GCSE Biology course. We're going to be revising organisation, which is the second big topic. So if you've always been confused by the circulatory system or dumbfounded by the digestive system, then you've come to the right place, because in the next 30 to 40 minutes we're going to be covering all that. Please subscribe if you find this useful so that other people can find it more easily, and we're going to go right over to the advanced information. So, first up, Combined Science Foundation 4.2.2, Animal Tissues, Organs and Organ Systems. This is going to be in everyone's exam, by the way. That's definitely coming up. So is the food tests practical, required practical. That's also coming up in everyone's exam. And as for stuff that's not coming up at all, the coronary heart disease stuff and all the cardiovascular diseases are not going to be examined at all for Combined Science Foundation, and the plant tissues and organs bit might come up. Combined Science Higher, once again we see the animals, tissues and organs bit, we see the food tests practical, but we also see the effect of pH on the rate of reaction of enzymes practical come up as well, and cut out entirely is the plant tissues, organs and organ systems. So Combined Science Higher, you can focus entirely on animals. Now everyone on the triple science course, once again the animal organisation bit, the food test practical, but taken out from triple science foundation as the principles of organisation. So that's going to be the very first bit that we do, as well as the bit on blood, which I'll mark out, and the bit on cancer. Triple science higher, once again we have the animals stuff. We also have the plant stuff, so triple science higher focus on both bits, and similarly to triple science foundation, blood and cancer have been taken out. Note for triple science, the food test practical is there, but not the effect of pH on enzyme reaction rate. So we're going to start with animal organisation. So we're going to look at cells, tissues and organs found in animals, specifically humans. But first of all, we need to look at the basic principles of organisation. So cells are the basic building blocks of all living organisms. Tissues are a group of cells with a similar structure and function. Organs are made up of groups of tissues performing similar functions. An organ system is a group of organs, and organisms, we're organisms, we're made up of organ systems. So we're going to have a detailed look at enzymes first off. So enzymes are biological catalysts, which means they increase the rate of chemical reactions inside the body without being used up themselves. Now, something you need to know about enzymes is they have this thing called an active site, and it's where the reactants bind to, and then they react. It provides them with an alternate reaction pathway. There's a bit of chemistry there, if you remember, and it just means they react quicker. So here we are, you can see the active site and the substrates, as we call them, the things that go in to be reacted, and each enzyme has a specific shape of active site, which can only fit a specific substrate. So we say that enzymes are specific. This word specific is very important in the exam. Enzymes are also proteins. So we call this model of the substrate going in, binding to the active site, we call this the lock and key model. And another thing to remember is that enzymes need the right temperature and pH to work. Too hot or acidic or alkaline, and this causes the active site to change shape, and then the substrates don't fit in and it doesn't work properly. And we say the enzyme has been denatured. And this very important word to remember. The chances of you being asked what denatured means is very high. And too cold, the enzyme does not get denatured, it just won't work because the conditions aren't warm enough. 37.5 degrees is usually optimum enzyme temperature. Anything above like 45 and it will probably have denatured. So now we're going to have a look at enzymes in digestion. Now digestive enzymes, they break down food into small molecules, and this means they can be absorbed into the blood. So that's why we have digestive enzymes. Now there's three main types. We have carbohydrates, protease and lipase. Obviously carbohydrates breaks down carbohydrates into sugars. Now we have a type of carbohydrates that we look at in detail, which is amylase, and this breaks down starch, which is obviously a type of carbohydrate. Protease breaks down proteins into amino acids, and lipase breaks down lipids, and remember lipids, they're just fats and oils, so lipase is for fats and oils, and these break down into glycerol and fatty acids. Now you need to know where these are produced. All of them are produced in the pancreas and the small intestine, as the small intestine is where this digestion happens. But protease is also produced in the stomach, which we'll look at in a second, and amylase is produced in the salivary glands. And another important thing, the products of digestion are used by the body to make new carbohydrates, proteins, and lipids. Some of the glucose is also used in respiration. So the two required practicals of this video are both to do with enzymes. So this first one is about the effect of pH on enzyme activity, and this is only going to appear in the Combined Science Higher exam. So if you're not taking Combined Science Higher, then skip to the next practical. The key thing to remember about this practical is we're going to be investigating amylase, which breaks down starch into a sugar called maltose. So the first step is using a pipette, put a few drops of iodine into each well of a spotting tile. Now, something to remember, this is going to come up in the next practical as well, iodine turns blue-black in the presence of starch, which is obviously why we're using it, so that we can tell when all the starch has been broken down by the amylase. So then we're going to set up the actual experiment. So we're going to place a test tube into a water bath set to 37 degrees, which is the optimum enzyme temperature. And why do we do this? Because the temperature is a control variable. We don't want our enzymes to denature due to too high temperatures or not work as fast. To that test tube, we're going to add amylase, and we're going to add a buffer solution with a pH of 5. And this buffer solution just simply sets the pH. It doesn't have any other effect other than changing the pH, which is obviously our independent variable. Now, then we're going to add starch solution, and we're going to start a stopwatch immediately and stir, and every 30 seconds we're going to sample it using a pipette and squirt it into one of the wells of the spotting tile and watch the iodine turn blue-black. And when we do this and the iodine remains orange-brown, doesn't turn blue-black, this means that all the amylase has broken down all the starch and the enzyme activity is finished. And then we're going to repeat it for different pHs and see how long it takes for all the starch to break down, and then we can calculate the rate of each reaction by doing 1 divided by time, and units are seconds to the -1. Now we have the food tests practical, which is coming up in everyone's exam, no matter whether you're doing combined science or triple science, which we're just testing for the presence of nutrients. So first, we need to get food as a liquid, so we need to prepare it. So we're going to grind it up. And this just means the whole sample is included. So if a nutrient is only in the very middle of the food, then we'll still get parts of it in the sample. We're going to add water to the ground up food, stir it, and this will hopefully dissolve some of the food if it isn't rock hard. And then we're going to filter it. We're going to remove the remaining solid bits of food so that hopefully we should have a solution of water and food. And now we're going to cover 4 different tests. First of all, to test for starch, like we just did in the other practical, we're going to mix with a few drops of iodine. Blue-black is the positive result. To test for sugar, this is the most complicated one. Remember, we're using Benedict's solution. These are all really important things to remember. For this one, we need to put it in a water bath, set it to 75 degrees C, and give it 5 minutes or so. And Benedict's solution, well, it's blue, so if it remains blue, then there's no sugar. If it goes green or yellow, that kind of means a bit of sugar, but the closer to brick red it is generally, the more sugar is there. To test for protein, we're going to mix a few drops of our solution with water and B-ray solution, and this will turn from blue to purple if protein is present. And finally, to test for lipids, we're going to mix a few drops of our solution with water and Sudan-free solution, and give it a shake to separate the lipid from the rest of the food solution. And the Sudan-free just acts as a stain, and it stains the lipids to the fats, remember, bright red, so a red layer will form on top if the lipids are present in the solution. So now we're going to have a look at our first organ system, the digestive system. Here we have a nice picture of a person where you can see all their digestive organs. And now the digestive system is responsible for breaking down and absorbing nutrients from the food we consume. It starts all the way in the mouth, which contains the salivary gland, which produces amylase to begin digesting starch. Remember, we covered amylase in the previous slides. Food travels down the esophagus to the stomach. This is also known as the gullet. And then we have the stomach, and this pummels food. Remember, it's got very muscular walls. It also begins to digest food. It contains enzymes such as pepsin. This is why we have stomach acid, because Pepsin needs a pH of 2, but stomach acid also kills bacteria that we may have eaten. So it's a really important defense mechanism. Now, the liver has various functions, but the one that you really need to remember is that it produces bile. And bile is important for two reasons. It gets released into the small intestine, this orange thing down here. First of all, it emulsifies fats, which just means it breaks them up into tiny droplets. This means they have a larger surface area, which means that lipase digests them quicker, because fats can sometimes be hard to digest. And it also neutralizes the stomach acid that can get into the small intestine, which makes sure, and it also makes sure that the conditions in the small intestine are alkaline, which is what many of the enzymes there need best to work. The gallbladder simply stores bile, and the pancreas, this produces digestive enzymes, this little thing here, and it releases them into the small intestine. And this small intestine is very important, as we covered in the last topic, it contains villi, and this is where the products of digestion get absorbed into the blood. And remember, the small intestine, as well as the pancreas, produces digestive enzymes. And then we have the large intestine over here. This absorbs water from the food, turns it into feces, and finally the rectum stores feces while the anus excretes it. So now that we've done the digestive system, we're going to look at the circulatory system, the process by which oxygen and other bits and pieces get distributed around the body, so that oxygen, for example, can go and be used for respiration in each and every cell in the body. We're going to start by looking at the heart, which is one of the organs in this system. We think about it as the center of the circulatory system, and it actually has two sides. The right side deals with the deoxygenated blood that comes in from the body, and the left side deals with the oxygenated blood once it's come back from its visit to the lungs. So the first step is the deoxygenated blood from the body enters through the vena cava, and then it goes through the right atrium, which is the top bit, the right ventricle down here, and they contract, they push it through and up here through the heart valve, and this just stops the blood flowing the wrong way, and it goes up to the pulmonary artery, and this takes blood to the lungs so it can be oxygenated, and the blood comes back through the pulmonary vein. And you can remember this because arteries leave the heart, so this is pulmonary artery, whereas the veins travel back, so this is the pulmonary vein. And then obviously this goes through the left atrium and the left ventricle. They contract, they push the blood through, up through the heart valve to the aorta, and this takes the oxygenated blood to the rest of the body to supply each and every cell with oxygen, as well as all those other essential substances we'll look at in a bit. It also has a pacemaker in the wall of the right atrium, and this produces regular electrical impulses, which causes the atriums and ventricles to contract, which pushes the blood through the heart and ultimately through the rest of the body as well. And with the blood going to the lungs as well as the rest of the body, we have a double circulatory system, as the blood does two loops. And with that, we're going to look at the lungs. Now, the lungs, similarly to the digestive system, are connected to the mouth through the windpipe, which we call scientifically the trachea. And this, connects the lungs. And when it reaches the lungs, it splits off into two tubes called bronchi. Each one is called a bronchus, but each of the bronchi splits off into lots of bronchioles, and these all connect to an alveolus, plural alveoli. So E is plural of us, general rule. And the alveoli, we looked at them in detail, how they exchange oxygen and carbon dioxide in the last video, which you probably go and refresh if you've forgotten. So it's all by diffusion, remember, they have all this one cell thick and these things to increase the effectiveness of the exchange surfaces so that oxygen diffuses into the blood and carbon dioxide as the waste product diffuses out, waste product of respiration, I mean. And then the diaphragm down here, this is a muscle that moves down to breathe in and back up to breathe out, in charge of contracting and expanding the lungs. And then we have this pleural membrane that surrounds both the lungs, and then the ribs protect the lungs and heart, because the heart is in the middle of the lungs, believe it or not, not on the left of the body, as many people believe. And between the ribs we have intercostal muscles which allow the chest to expand so that we can breathe in. It would be pretty disastrous if we didn't have them, really. So the final piece of the puzzle in the circulatory system is the blood and the blood vessels. So here are 3 blood vessels, completely accurate drawings. The arteries, first of all, they carry blood away from the heart, like we looked at earlier, and they're thick, really thick walls, and they're strong because the blood is being pumped with a high pressure out of the heart to the rest of the body, and they have this thick layer of muscle as well, which enables them to stretch to allow the blood through. And then we have the veins, which carry the deoxygenated blood back to the heart. They have a large lumen, and this is the inside of the heart. They call it the lumen. What an awesome word. This helps the blood flow at the low pressure, because obviously it's got to flow, I don't know, up your legs, back into the heart. And they also have heart valves to keep the blood flowing in the right direction. And then this tiny little one down here is the capillaries, and capillaries are so important. So when you think of us exchanging substances between the blood, it's always between the capillaries. The arteries branch off into the capillaries, which reach every cell, which is why they're very small, 1 cell thick, and this also increases the rate of diffusion. And then the capillaries, once they've exchanged substances, their oxygen, their whatever, with the cells, then they go back into veins and back to the heart, and they also have permeable walls to allow substances to diffuse out. Now, this next bit on the parts of the blood is not going to come up in the triple science combined or higher exam. It's only for the combined science people. Blood is a tissue, and it's made up of 4 different main parts. So you have the red blood cells, which are responsible for transporting oxygen because they contain hemoglobin, which oxygen binds to. This is red pigment, which is why they're red blood cells. White blood cells, this is all about defense. We'll cover them a bit more in the next video, and they can engulf harmful bacteria. This is called phagocytosis, and they can also produce antibodies and antitoxins, which again we'll look at in the next video. Platelets are small fragments of cells with no nucleus, and they help blood to clot at wounds and prevent infection. And plasma, the blood plasma, that's just the liquid component. It carries everything. It carries the red blood cells, white blood cells, platelets. It carries the glucose and amino acids, the product of digestion. It carries urea to the kidneys. It carries the hormones around the body. It carries carbon dioxide back to the lungs. It carries proteins. It's so much more. It carries the antibodies, antitoxins. Plasma just carries everything. So now we've looked in detail at the circulatory system, we're going to have another look in detail at all the cardiovascular diseases that can arise, any problems with the heart, starting with coronary heart disease. And this is a type of cardiovascular disease where the coronary arteries, and these are the ones that supply the heart with blood, and they get blocked by a build-up of fatty substances, fatty deposits, could be cholesterol or other. And this leads to a restricted blood flow, which can lead to heart attacks and other nasty symptoms that are life-threatening. And we have to look at a few ways of treating different cardiovascular diseases. We're going to start at ways we can reduce the risk of CHD, which I'm going to call coronary heart disease. We're going to start with stents, which are tubes that we can put into arteries to keep them open and squish fatty deposits against the walls. And we have to look at the advantages and disadvantages of each treatment. So the advantage of stents, they reduce the risk of heart attacks for people with CHD. They're also more long-term. They don't trigger an immune response from the body, unlike heart transplants, for example. However, it's an operation, so there's risks of infection, there's risks of heart attacks, blood clots are possible. And then we have statins. Statins are drugs that you can take. They lower the cholesterol in the blood by stopping the liver or lowering the amount that the liver produces. Too much of a certain type of cholesterol can lead to the fatty deposits that cause CHD in the coronary arteries. Now, the advantages, again, they reduce the risk of heart attacks and CHD. And some studies have also shown that statins can reduce the risk of other diseases, like Alzheimer's, for example. But a disadvantage is they have to be taken regularly, so unlike stents, they're not a long-term solution. And there's also quite a number of negative side effects, like headaches, feeling faint, and they can be serious. Liver damage, kidney failure is a possibility when they're taken regularly, so that is a big disadvantage. And now moving on from CHD and onto general cardiovascular diseases, heart valves, they can become damaged by infections, or old age can make them weaker. And they can either become harder, which prevents them from opening properly, or they could the opposite happens. They become looser, they allow blood to flow the wrong way. Either way, this means that blood doesn't flow around the body properly, which is life-threatening again. And we have two ways to replace faulty heart valves: biological valves, which are taken from other humans, or mechanical valves, which are man-made, obviously. But mechanical valves, an advantage of them is that they wouldn't be rejected by the body. Advantage of biological valves is that they would work better than mechanical ones, maybe. So an advantage of these heart valve replacements as a whole is that they're less risky than replacing the whole heart, but there are still risks of complications such as blood clots, as there are in any surgery whatsoever. Now we're going to look at heart failure, which is very serious. Heart transplants using donor hearts are usually used. However, there's not usually a good supply, so sometimes we use artificial hearts. We'll have a look at them in a minute. So donor hearts work better than artificial hearts, and they're often a more long-term solution, as you know, you can keep them in as long as the immune system doesn't reject the new heart, as it recognizes the new tissue as foreign, and that's always a risk, a big disadvantage. And this could mean that the person has to go on immunosuppressants, which can make them more susceptible to other communicable diseases. And CHD, by the way, is a non-communicable disease. We'll look at what that means in the next video, or actually in this video, I think. And artificial hearts, they're just mechanical hearts that are used to pump blood around the body when your own heart cannot. The advantage is they won't be rejected by the body, unlike donor hearts, and they can also be used as a permanent fix sometimes. However, there are disadvantages. The artificial heart could fail, like the electric motor could stop working and your heart could just stop. That would be a problem. And they can lead to blood clots and strokes, as the blood doesn't flow through them as well. And this means that the patient, instead of having to take immunosuppressant drugs like donor hearts, they'd have to take blood-thinning drugs, which means that they really can't risk many accidents, otherwise they'll just keep bleeding, no blood clots will form. Finally, artificial pacemakers are devices that can be implanted to help people with irregular heartbeats, and like natural pacemakers, they produce an electric current, and they can help people, again, with irregular heartbeats. They reduce the risk of heart attacks by keeping the heart beating regularly. However, there's a risk of infection during surgery. And then I've got a memory test for you, because obviously we've covered a lot in the last 10, 12 minutes or whatever. So pause the video, have a go at these. So a catalyst is something that increases the rate of a chemical reaction without being used up itself. An enzyme is denatured if the active site has changed so much that it can no longer catalyze the reaction that it is meant to. Amylase is produced in the pancreas and small intestine, just like all the other enzymes, but it's also produced in the salivary gland. And we have stomach acid to kill bacteria and provide the conditions for enzymes like pepsin to work. And deoxygenated blood goes in through the vena cava on the right side, and the pulmonary artery takes it to the lungs to be oxidized. So a pretty big focus for the biology paper one is health issues and health and disease. So now we're going to have a closer look at health issues. We've already looked at one type of disease, cardiovascular disease, and the treatments to it, but now we're going to look at health issues in general. So the definition of health is the state of physical and mental well-being. That's very important. Both physical and mental. Diseases can cause ill health, obviously. There's two categories of diseases. In the next video, we're going to look in focus at communicable diseases. They are the infectious diseases that spread between people, like the flu, COVID, etc. Non-communicable diseases, for example, cardiovascular disease, coronary heart disease, they cannot be passed between people or animals, like cancer, again. And we're going to have a closer look at these now. But first, we're going to look at how different diseases, communicable, non-communicable, physical, mental diseases, they can interact. There's 4 different ways that you might be asked about. Diseases that damage the immune system can leave a person more vulnerable to infectious diseases. So you have maybe a non-communicable disease that damages your white blood cells or something. That makes you more vulnerable to communicable disease. Viruses, so a communicable disease can sometimes cause cancer, a non-communicable disease. An immune response to a pathogen, so that would be a communicable disease, that can trigger an allergic reaction or worsen asthma symptoms, non-communicable again, and damaging physical illnesses such as cancers can affect people's mental health, especially if they know their life expectancy is decreased, so a link between physical and mental health there. And the factors that affect a person's health. Now, the main factors are the healthy, balanced diet and exercise, so your physical fitness. Again, that's going to protect you from a lot of things. We'll look at that in a minute. How stressed a person is, because you may well know that stress can cause many complications, especially if you're regularly feeling very stressed. Or a person's life situation. So, for example, can they access healthy food? Can they access condoms to prevent STDs? Now, we're going to look at risk factors and how they can increase the likelihood of non-communicable disease. So risk factors are things that increase the likelihood of a person having a disease. A causal mechanism is something that causes something else. So hopefully you would never have to see this word again, but just in case you do. So first of all, smoking is a big risk factor for lung disease, lung cancer, cardiovascular disease, because it damages arteries and cells. Smoking is very damaging. Don't smoke while pregnant, because it can cause health problems for unborn babies. A poor diet and lack of exercise, again, we saw this as a factor that can affect a person's health. This can lead to obesity, which can increase the likelihood of type 2 diabetes and cardiovascular disease. Drinking alcohol can lead to liver disease, damaging nerve cells, and similarly, drinking alcohol can damage unborn babies if you drink it while you're pregnant. And carcinogens, these are simply substances that can cause cancer. For example, ionizing radiation from X-rays, UV light from the sun, that can increase the risk of cancer, which is a non-communicable disease. And this is quite a scientific section, so you need to remember that correlation between two things does not mean that one has caused the other. We have to prove every single one of these scientifically before we can just say it. And finally, the distribution of non-communicable diseases around the world. People from more developed countries, interestingly, are more at risk of non-communicable disease due to the fact that they can simply afford more food. So they might have a worse diet if they're going to McDonald's every day, enjoying lots and lots of rich, high-fat, high-salt foods. They don't have to do as much exercise, and that can all lead to obesity and much higher obesity rates in higher income countries. And yeah, it leads to more non-communicable disease. And nationally in the UK, people from more deprived areas, they have a worse diet. I don't know if you've ever been shopping and noticed how much more expensive the fruit and veg is compared to the chips and the chocolate. So people from deprived areas can suffer from a poor diet, and therefore things like cardiovascular disease are more common. Now, non-communicable diseases have costs, quite obviously. We can split them into two factors. The first three are human factors, the second three financial factors. So human factors, for example, they cause millions of deaths worldwide, they decrease a person's lifespan and quality of life, and they affect the sufferer's family and friends. Those are the three human costs. And the financial costs, again, the cost of treatment, the funding going to the NHS, that's huge. The cost of living with a non-communicable disease, if you need adaptations to your lifestyle, that can be another cost. And non-communicable diseases decrease the number of people able to work, which has an economic impact. That's a third financial cost of non-communicable diseases. And finally, as I mentioned a minute ago, this is a very scientific section. So around this point, you're very likely to be presented with a graph with data from non-communicable diseases. It could be a bar chart, histogram, scatter graph, any graph you might have seen before from maths. And you should also be aware of the principles of sampling, so how scientists collect that data for the graph. You could be asked about that. So scientists study a population by taking a sample of people, and these people have to be representative of the population as a whole. And this means there can't be any bias, so we have to have 50% men, 50% women if we're looking at people in general. People's age has to be equally distributed, so does lifestyle. These should be taken into account. And also, the broader the sample is, so the more people there are, the more accurate the results will be. and accuracy in science is how close to the true value something is. And finally, combined science only thing. This isn't coming up in either triple science exam. We're going to look at one final non-communicable disease, which is cancer. And cancer is a result of unwanted changes or mutations to cells, which then divide and grow uncontrollably into a tumour. That is the definition of cancer. Learn that, memorize that. And there's two types of these cancer tumours. The benign tumours, they're actually non-cancerous. These are the good ones. They stay in one place. Well, I say good, better than malignant. A tumour is not good. They're often inside a membrane. They don't spread to other parts of the body, which means they're much more easily treated. Malignant tumours are cancerous. They're dangerous. They can often be fatal because they spread around the body through the blood. They spread to healthy tissues anywhere in the body, and they form secondary tumours. They can be very hard to get rid of, and again, possibly fatal. And now, risk factors. Always remember your risk factors for non-communicable diseases. So smoking up here is the top one. It's been linked mainly to lung cancer, but also to many different other cancers. So smoking is just really bad for you. Obesity is a risk factor of many different types of cancer. Again, obesity, smoking. You can see these are general risk factors for non-communicable disease. Exposure to UV radiation. You may have been told to wear suntan lotion when you're outside in the sun on the beach. This is why, because it increases people's chances of skin cancer, because UV radiation can be ionizing, and certain viruses could increase the likelihood of a person developing cancer. You'll remember that was one of the ways different diseases interact. And finally, you can inherit genes which make you more likely to develop cancer in your lifetime. The fact that you can inherit these more cancer-prone genes, that's very important to remember as well. So we've made it to the very end of animal organization. There's a lot to remember there, so here's 16 questions to test yourself. Pause the video, have a go. So an enzyme's active site is where the substrates bind to. Remember, it's specific, and lipase breaks down fat. Pepsin is produced in the stomach. Bile emulsifies fat, so it has a higher surface area so that lipase can break it down more easily, and it neutralizes stomach acid in the small intestine so that the enzymes there can work more effectively. The large intestine absorbs water from food to form feces. The pulmonary vein carries oxidized blood back from the lungs to the heart so that it can be pumped around the rest of the body. So blood, deoxygenated blood, comes through the vena cava into the right atrium, down into the right ventricle, up through the heart valve, through the pulmonary artery to the lungs, back through the pulmonary vein, into the left atrium, down into the left ventricle, up through a heart valve, and through the aorta, which takes it to the entire rest of the body. We need intercostal muscles. They are between the ribs so that the chest can expand so that we can breathe in. The vein has a large lumen, remember that bit in the middle, so that blood can flow at low pressures more easily, and it has heart valves so that blood can flow the right way. This is a combined science only question. Platelets are small fragments of cells that help the blood to clot. Long-term method of treating coronary heart disease is stents, because statins, they're kind of the pills that you have to take regularly, but stents, they're just fitted in those tubes and they stay there forever. They work forever, or long term, not necessarily forever. But an advantage of an artificial heart, again, it won't be rejected by the body, unlike a donor heart. But however, it can it's also more readily available, but it can break. For example, the electric motor can fail, and also blood doesn't flow through it as easily, which means it's more likely to clot, which means the patient has to take blood thinning drugs, which can also have side effects. Non-communicable disease is a disease that doesn't spread between people and animals. So, for example, CHD, cancer, etc. Bad physical health can cause bad mental health, especially when you know your life expectancy is decreased. Again, you can have human or financial costs for non-communicable diseases. So human might be that it kills millions of people. It affects the quality and length of a person's life, and it affects the sufferer's friends and family. Financial, the cost of treatments, that the NHS has to pay is huge. The adaptations, the cost of adapting to living with a non-communicable disease, again, can be quite big depending on the disease. And non-communicable diseases mean that less people are able to work, which can affect the economy. And if someone is obese, it can put them at a higher risk of type 2 diabetes, cardiovascular diseases, and cancer. So now we're going to look at plant organization. If you're combined science higher, you can skip to the end. And this is going to be a major focus for the triple science higher exam. So the structure of the leaf we're going to look at first, because there's lots of important plant tissues in there. Most plants have four main organs. The roots are an organ, the stem is an organ, the leaf is an organ because it's made up of all these tissues, and again the flower or the reproductive organ is another organ. So the meristem tissue is the tip of a plant's shoots where growth happens. We saw that in the last video. And now we're going to look at all the tissues in the leaf. So on top and the bottom, we got the waxy cuticle to help protect against water loss. And the first layer of cells is the epidermal tissue to protect the leaf, and it's also transparent to allow light through, to the palisade mesophyll layer, and these have lots of chloroplasts, these cells, to carry out lots of photosynthesis, because if you remember, these contain chlorophyll, a green pigment which absorbs light for photosynthesis, which is why these are transparent. So this is also near the top of the leaf so that it can absorb more light. The spongy mesophyll layer, this is also meant to carry out lots of photosynthesis. The air gaps, as we saw again in the last video, this allows diffusion of carbon dioxide and water into the cells to be more efficient. These lower epidermal cells have a similar function, but they also have gaps called stomata, which allow the carbon dioxide to diffuse in, and they've got the guard cells either side, again, which we'll look at more detail in a bit. And xylem and phloem tubes, again, we saw these in the last video. They connect to the leaves, they supply food and water, and xylem supplies water, phloem supplies the food. That's very important to remember. So then we're going to look at the plant transport systems, xylem and phloem, in a lot more detail. So the phloem tubes, these transport the food, the dissolved sugars, through the plant. They are living cells with small gaps in the end. That's their structure. And this process by which they transport the plant's food from the leaf to where it needs to be stored, this is called translocation, and it happens in both directions. That's important to remember. Now, xylem tubes transport the water and minerals from the root hair cells, which have their large surface area to increase the rate of diffusion and active transport. These absorb the water and minerals, and they then travel up the xylem tubes through the stem to the leaf for photosynthesis. And also some of the water evaporates out of the leaf through the stomata, and this process is called transpiration. And the structure of xylem tubes, they're made from dead xylem cells. Phloem tubes made from living phloem cells. Xylem tubes use dead xylem cells with holes in the ends, and we've seen they're strengthened by lignin, this strengthening substance. So we're going to have a look at transpiration in more detail. We've got factors that affect the rate of transpiration, this rate of water loss as the water evaporates out of the leaf. A warmer temperature means a faster rate of diffusion of the water out of the plant, which means that transpiration happens faster. Again, these are mainly the rate of diffusion because the water diffuses out of the plant through the stomata. The brighter it is, the more the stomata open to allow the CO2 in, so the more transpiration happens. The drier it is, the higher the concentration gradient of water, so the more transpiration, and the more airflow, the greater the concentration gradient, so more transpiration happens. In the lab, we use a potometer to measure the rate of transpiration. And now we're going to look at how the stomata are adapted to minimize this water loss through transpiration, and the guard cells. Now, the guard cells, they sit on either side of the stomata. When the plant is short on water or it's dark, they lose water. These cells, the actual cells, they lose water. They become flaccid, and this means that they naturally they lean in towards each other and close up. And when the plant has lots of water, the guard cells absorb the water, they become turgid, they open up the stomata, ready to allow CO2 to diffuse in, and the stomata are on the underside of the leaves. It's cooler, has a lower light intensity, and these are all adaptations to reduce water loss through transpiration. So we've made it to the end of biology topic 2, cell organization, and if you watched to the end of my previous video, you know that how I like to advise people to revise is through lists of questions, because you test yourself through active recall. You come back the next day and test yourself again on any that you got wrong, and then in a week, or depending on how close it is to the exam when you're watching this, you can come back and test them all again. So you can either test them in your head, you could write them all out. I find that testing in my head is easier, but generally you have to remember you have to remember 20 different answers when you go back through the video and look at the answers, so writing them down might be better. There's some bits from different topics that are in different exams and aren't in different exams, so I have colours here telling you which questions are for you, which questions aren't. The black questions are for everyone. So on this slide we have the enzymes and digestion questions. So these four magenta questions, they are not for Triple Science Foundation exam, but in all the others they could come up. And this question 15, this is the required practical that's only in the Combined Science higher exam. The circulatory system questions, these orange questions, they're only for the Combined Science higher and foundation. They will not come up in the Triple Science higher and foundation exams. The non-communicable diseases questions, these purpley blue ones, they are not for Combined Science Foundation, so that's all of the coronary heart disease questions. And the cancer questions are the same orange because they are Combined Science higher and foundation only, and not for Triple Science. Finally, the plant organization questions, Combined Science higher, you don't have to worry about any of these at all, they won't come up, and these will be a focus in the Triple Science higher exam. So I hope everyone found this helpful, and that if you did, you could subscribe so that other people can find this more easily, and that you have a good time revising and get a great grade in your exams.